Hello everyone, this is Glennis May Garcia. Today we look at general process of mitosis and meiosis in this page. We're going to add some extra detail including the names of the stages, and you will learn on how mitosis and meiosis work. Mitosis is a process where a single cell divides into two identical daughter cells. It is a process of cell division that uses identical copies of cells, and it involves in growth and sexual reproduction. When cells divide by mitosis, the number of cells increases and hence the organism grows. Different organisms have different numbers of chromosomes. You could keep it simple, just going to look at what happens to one chromosome during one cell. Before we start to the stages of mitosis, just a quick reminder that a chromosome is made up of two chromatids, and one from the mother and one from the father. Interphase. This is when the cell grows and copies its DNA before moving into mitosis. The chromosomes duplicate and become two identical chromosomes, joined at the centromere. So in humans, it has gone from original 46 to 92. Prophase, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. In prophase, in the nucleus, the chromosome condenses and in the cytoplasm spindle fibers form. The metaphase. The nucleus disappears and the chromosome appear in the cytoplasm of the cell. When nuclear membrane breaks off, the spindle fibers attach to the chromosome, and the chromosome line up at the equator of the cell. Anaphase, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. In this stage, the spindle fibers shorten and the centromere divides so that each chromosome becomes two separate images. Telophase, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. The nuclear membrane moves from each set of chromosomes. Chromosomes spring back oscillate, new ups, and the spindle fibers break down. In humans, each nucleus has a normal 46 chromosome. Cytokinesis. The cell membrane pinches in at the cell equator, forming a cleft called the cleavage furrow. Cell membrane pinches in the two separate sets of chromosomes into the identical daughter cells, with the same number chromosome as parents, so 46 or 23 phase in humans. They will have now cells divide by mitosis to form two new identical cells. Let's go to meiosis 1 and 2. Many organisms has their genes to their offspring through sexual reproduction. This begins when two gametes unite to form an embryo that is genetically unique from the parent organism. The embryo then grows into an adult who in turn passes their genetic information onto their own offspring. Gametes are formed through a process called meiosis. In meiosis 1, the prophase 1, the DNA condenses to form chromosome. Here we see the duplicated sister chromatids joined together at the centromere. These stay fused at the centromere throughout meiosis 1. Next is Metaphase 1. Here, the synapse chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations. Each time meiosis occurs, that's it. Anaphase 1. During this stage, homologous chromosomes separate and migrate to the two poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final step of meiosis 1 are Telophase 1 and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis 2. Meiosis 2, prophase 2. Again, chromosome contains the nuclear envelope breaks down, and the spindle's apparatus forms. The major difference between prophase 2 and prophase 1 is the fact that the daughter cells only hear one copy of each homologous chromosomes. So in prophase 2, there is no synopsis of homologous chromosomes or crossing over. Metaphase 2. The chromosome align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for this chromosome to align. In anaphase 2, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed further apart as microtubule elongate. In telophase 2, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis 2 began with two cells and each of these cells were split into two cells, we now have four unique haploid cells at the end of meiosis. These cells are gametes. That will be all. Thank you.